All right, welcome back. In this short lecture video, we're going to introduce skeletal muscle tissue. So let's talk big picture. We know that we have certain functions that our skeletal muscle system carries out. So for one thing, along with our bones, right, the skeletal muscles help us move our body. Large movements, small movements, from throwing a ball to blinking my eye, those are controlled by muscles. Our posture is maintained by muscles. Muscles help to form protective barriers and support our internal organs. We have circular openings that are sphincter muscles that regulate the elimination of material from our digestive tract and our urinary tract. And because muscle contractions use energy, they're going to produce heat, and so it produces heat and helps us to maintain our body temperature. All right, let's look at the gross anatomy of muscle tissue. So first of all, a muscle cell is many times called a muscle fiber. So those are two terms that mean the same thing. And when you look at the shape of a muscle cell, we understand why it makes sense to call it a fiber. So a muscle can be long, and a muscle cell is going to be the entire length of the muscle. So it's this long linear structure. Now in development, cells come together and can fuse to become this single muscle fiber, and therefore we have more than one nucleus per muscle cell or muscle fiber. So we call these multinucleated because they have more than one nucleus. Now, if we look at, so we can look at one muscle cell, right, or one muscle fiber here, and we see that those are bundled together in a group, and they're covered by a protective covering, and that is called a fascicle. So a group of muscle cells or muscle fibers is called a fascicle. Then we have multiple fascicles grouped together, covered by some connective tissue that make up the muscle itself. Now, let's talk about these layers of connective tissue. So the internal layer, the layer that wraps around each individual muscle cell or muscle fiber, is the endomysium. That makes sense, right? Endo would be internal, so that's the most internal connective tissue layer. And then we have around the fascicle, we have the perimysium. So finally, around the muscle itself, we have the epimysium. Epi meaning above, right, or on top of. Now notice that the epimysium continues, and we see that the muscle then is going to end with either a tendon that's going to connect that muscle to bone, or an aponeurosis, which may connect a muscle to deep fascia or something else that's not a bone. All right, let's zoom in a little more and look at what this muscle fiber or muscle cell looks like and what are the components of a muscle cell. So we know something already about cells. We know that all cells have a plasma membrane that surrounds the cell. That's what separates what's inside the cell from outside. And skeletal muscle fibers have this division, but we give it a special name. So the plasma membrane or the cell membrane of a muscle cell is called the sarcolemma. That will make sense to you in a minute when we learn that the contractile unit of a muscle fiber is called the sarcomere. So when you see the word sarco, we're going to think of contraction or a muscle cell or muscle tissue. So the plasma membrane is called the sarcolemma. Muscle cells need lots of mitochondria because if you'll recall from when we learned about cell organelles, the mitochondria are the powerhouse. So they are the generator of energy, and as muscles contract, they use a lot of energy, so we need a lot of mitochondria. Now remember that cells have something called an endoplasmic reticulum, and in a muscle cell there is a specialized endoplasmic reticulum called the sarcoplasmic reticulum. And when in a future video when we talk about the actual muscle contraction and how calcium ions are released, which causes the muscle to contract, we will look specifically at how that happens in the sarcoplasmic reticulum. Now, I mentioned that there are multiple nuclei for a particular muscle fiber. We have something else that's special to a muscle cell called a T-tubule or a transverse tubule. So these, as you can see, are basically invaginations, right, into the interior of the muscle cell. And this is very important as we look at how muscles contract. Part of it is going to be because of electrical signals traveling. So these T tubules, these infoldings into the cell, are going to allow signals to travel to all parts of the muscle cell or muscle tissue. Now notice, and we'll talk about this in more detail, but the T tubules are going to be present near the sarcoplasmic reticulum. And that's going to be important when we talk about the release of calcium from the sarcoplasmic reticulum. One more feature of a cell that I'm going to mention that's not actually on this particular figure is we know that all cells have to have cytoplasm, right? But in a muscle cell, we actually call that sarcoplasm. So there's that word sarco again. So the cytoplasm of a muscle cell is called sarcoplasm. Now I mentioned to you that the contractile portion of a skeletal muscle is called a sarcomere, and we're going to look at this. So this is basically all along this muscle fiber, all along this muscle cell, are these repeating units that we call sarcomeres. So as we see this muscle cell, right, muscle fiber, we can see a part of it extending. We can see these repeating units that are flanked by these blue lines. Okay, in between each of those blue lines is what we call a sarcomere. And the reason these sarcomeres exist is based on the way that two different protein filaments or myofilaments come together in the muscle fiber. So these two filaments are myosin, which is called the thick filament, and actin, which is called the thin filament. So these are arranged in a particular way. Each sarcomere, so if we look at these repeating units, right, the beginning and end of a sarcomere occurs at these Z discs. So notice that they're kind of zigzag, right? They're not straight lines. So the Z disc is going to be sort of the marcation of each individual sarcomere. And remember, these are just repeating all along the entire muscle fiber or the entire muscle cell. Now, there are some components of the sarcomere that I want us to recognize. So remember when we looked at a microscopic image of skeletal muscle, one of the things we said is it has striations, right? It has this striping pattern. Well, the striping pattern that we see under the microscope comes from the way these filaments are arranged in this sarcomere. So we have different what we call bands that are either lighter or darker in the sarcomere, and that's what causes the banding pattern or the striation of the muscle tissue, and we can see that under the microscope. So in this image, what I want to point out is the Z discs, right? That's the marcation, right? That marks where the sarcomere is. In the very middle, between the two Z discs, is the M line. That's easy to remember, right? The middle line is right in the middle. And then in the next slide, we're going to look at some particular zones that we name these zones based on how they appear under the microscope. So if they have more of the thicker filament, they're going to stain darker. If they only have the thin filament, they're going to stain lighter. So we're going to point out the A band and the I band on the next slide. All right, here's our sarcomere. And the first thing I want to do, so this, right, is one Z disc, and this is the other. So we're going to focus on the sarcomere that is in between those two Z discs. Now, first of all, we have the myosin or the thick filament, and then we have the actin or the thin filament. So in this case, the thick filament is red and it's thicker, it's wider, right? And the thin filament is represented by blue. So that's our actin filament. So I want to point out we still have this M line, right in the center, but we have a banding pattern. So down below, we can see here's the division, right, of the sarcomere that we're looking at. But we see it's just a repeating pattern all along this muscle fiber. Now let's look at some banding patterns. So the portion of the sarcomere that contains the thick filament, the myosin, it's going to stain darker. It gives us a darker band, and that's called the A band. So notice that all along the portion that has the red or the myosin filament, that's called the A band. The portion that doesn't contain any of the myosin, that only has the actin, those are called the I bands. Okay? So the darker portion would be the A band, and the lighter portion would be the I band. Now, there's one other region that I want to point out to you, and that's this H zone. So what's different about the H zone from the rest of the A band? Well, in the H zone, so right now, I want to point out this is a sarcomere when it is relaxed. In other words, when the muscle or the sarcomere is not contracting. So when it is relaxed, right, in the H zone, we only have the myosin filament. We don't have any of the actin filament present. Now, the way that a muscle contracts, each of these sarcomeres is going to contract. And when they contract together, that's what causes the muscle to contract and makes the muscle smaller, right? It makes it does the opposite of lengthen, right? It shortens the muscle. The way that's done, and we'll look at this more on the next slide, is called the sliding filament model, or that's our explanation, right, for how this is working. And what that means is none of the filaments themselves, neither the myosin or the actin, are actually getting smaller, but they're sliding past one another, which shortens the sarcomere. So we're going to look at a little bit more about the filaments, and then we'll look at how this sliding filament model actually works. All right, so here's a close-up look at our two filaments. So I mentioned to you that the thin filament is the actin. That's the primary protein that's in the thin filament. However, we do have two other proteins, and we're going to talk about these much more when we talk about the physiology of how the contraction occurs. These are regulatory proteins called troponin and tropomyosin. Essentially what they do is they control whether or not the thick filament, the myosin, can bind to the actin filament. It just so happens that these are controlled by calcium ions. Okay, and we'll look at that in detail in a future video. The thick filament, the myosin, has these heads as part of this protein. Okay, these heads are what have the potential to bind to the actin filament. So if you look with me down here below, we can see the heads showing on the myosin filament actually binding to the smaller actin filament. Okay, again, we'll look at the details of how that happens, how the contraction happens in a future video. For now, I want you to be familiar with the two proteins, myosin and actin, and we're developing an understanding of the sarcomere and the banding pattern of the sarcomere. All right, this is our final slide to look at in this video. And what I want you to see is how the banding pattern and how the sarcomere changes when it goes from relaxed to contracted. So at the top, we're beginning with a relaxed sarcomere. Okay, and then below, we're seeing a contracted, or it may not be fully contracted, but it is contracting, right, sarcomere. Now, there's a lot to take in on this slide, but the first thing I want you to see is in this particular slide, the myosin proteins are purple, right? They're the thick filaments. And the actin proteins are green. We still have our banding. So everywhere between that, where the myosin proteins are present, that's the A band, that's the darker band, okay? The area where there is only actin and no myosin, those are lighter, those are the I bands, okay? We have our Z discs, right, which are those zigzaggy lines, so that marks the beginning and end of each sarcomere. We have our M line in the very middle, and the H zone is that portion, right, that doesn't have any actin. It's only the myosin, the thick filament. Now remember we said that the filaments themselves are not getting shorter when the sarcomere contracts. The filaments are the same length. What happens is the filaments are sliding past one another to shorten the overall sarcomere. So when it's contracting, here are our Z discs, or our Z lines. Notice they've moved in, right? They've moved in just a little bit, so we have a little bit of a contracted sarcomere. But the length of the actin and myosin are the same. What's different is the myosin heads have grabbed a hold of the actin protein, and they have moved the actin toward the M line, or toward the middle. So in the relaxed sarcomere, we had all this space in the H zone where actin proteins were not present. Now in our contracted sarcomere, our H zone, the actin has moved into the H zone toward the middle. So let's think about how this changes. So our A band, right, is the same exact length that it was. The A band has not changed, right? Because the A band was the length of the myosin, or the thick filaments. They didn't move. That hasn't changed. The I band, however, has gotten shorter. That's not because the actin molecules or the actin filaments got shorter. They didn't. They just slid toward the middle. So the region where we only had actin and no myosin got smaller. So as this sarcomere contracts, when it contracts fully, it will continue to slide where you have really no visible I band at all as those actins move closer and closer to the middle. Now, the H zone, right, has disappeared as the actins shift in. So the A band stays dark. We don't see a change there. The lighter I band is going to get smaller. Okay. Our M line has stayed in the same exact place. So just as summary, right? These bands, the reason we're talking about these bands is this is what we actually see under the microscope when we see the striping or the striation pattern. What causes the bands is which particular protein or myofilament is present, whether it's myosin only, myosin and actin, or actin only. And the filaments themselves are not shortening, right? It's the sarcomere is shortening as the filaments slide past one another. And that's called the sliding filament model.